Today, we're going to be talking about our motility medium, which is a differential medium used to differentiate bacteria based on, well, motility. So the basic way that our medium is set up is that it has a 0.35% agar concentration. It makes it a nice semi-solid medium, kind of like you can see, it doesn't fall down when I tip it over. And that 0.35% agar concentration will prevent the spread of non-motile bacteria throughout the tube, so you only see growth along the stab line. Whereas motile bacteria will be able to spread all throughout the tube, and it will sort of turn the whole tube cloudy. Another thing to note with our medium is that sometimes it's made with tetrasolium, and tetrasolium will basically turn any area where there's growth red, making it a little bit easier to interpret our tubes. Another thing to note on the media is that since it is a semi-solid media inside of a tube, whenever bacteria grows, they tend to produce a bunch of gases, and those gases can get trapped inside the tube and inside the media, either breaking the media or breaking the glass itself. So normally we tend to grow these at 25 degrees Celsius rather than 37 degrees Celsius to help limit some of that gas production. But kind of with that out of the way, let's talk about results. So after you've incubated them for your 24, 48 hours or a week or so, depending on how long we're doing it, and we're coming back to score them. So if you have growth, again, only along the stab line, like you can see here, that is going to be a negative result for motility. But any deviation away from the stab line, kind of like in these guys here, so even if it's just this little bit of deviation or this very well-defined positive result here, any deviation is going to be a positive result. You might sometimes even see that just the outside edge of the tube starts getting really, really red as well. That's also going to be an indication of the positive result. Now, these three tubes here were made with the tetrasolium, which is why they're looking really red. But I have an example here of some that do not have it, so we can look at what that looks like too. So in this one here, you can see, again, just nice growth down along the stab line here, but most of the tube is fairly transparent. You can see through it very well. Whereas on this one here, it's very cloudy all the way throughout. So again, that's going to be, this one is negative for motility. This one is going to be positive for motility. Now, in terms of actually inoculating our tubes, we have to use the stab technique. And remember, in our stab technique, we have to use a needle, not the loop, in order to do it. And generally, when we do a stab, it tends to be better from a broth. So a broth to deep stab technique is generally better. But you can also do it if you do it from either a plate or a slant. It just generally turns out better from a broth. But that should cover what we need to know about our motility medium.